My name is Rob GULDEN, and I'm with the Department of Plant Science at the University of Manitoba. And thank you for the opportunity to talk about this project, which we started roughly about a year ago. And the project revolves around modeling seed protein content in canola. And I've listed it as part one. We're early into the project, and we're going to look at sources of variation that are going to help us, in the end, develop a better model. This is a slide of the partners involved with the model in this project. The project's being funded by the Protein Industries Canada conglomerate, who are interested, obviously, in increasing plant protein and the market thereof. Myself and Jonathan Rothet are at the University of Manitoba, and Jonathan is doing most of the modeling. He's in my group as a research associate / technician. Other partners involved are Corteva Agroscience, principally Chad Casilny and Dave Charney, but also their entire modeling group, and we've been working with those on some field studies that I won't be talking about today. And third, we also have Veronique Barthé involved in this project from the Canadian Grains Commission, and they and Corteva have provided us with some data sets that we are analyzing. So what are we trying to accomplish with this project? Well, we're trying to figure out protein content and to some degree oil content as well, but I'll only speak on protein content today. And the image on the left shows the relative proportion of oil and protein in the canola seed. And oil is usually around 45%, give or take. Protein is somewhere in the low 20 to mid 20% range. And of course, there's some variation associated with that, which I've highlighted with the brackets. And the question for us in this project is really, what are those sources of variation, and how many of those can we control, how many of those can't we control, and can we model that? So we're working with a relatively small sliver of total protein content and total oil content. The images or the graphs on the side show the oil content and protein content of the Canadian canola crop as determined by the Canadian Grain Commission over a period of 20 years. So we do see a fair bit of variation, about 4% in oil content and protein content, and this is the average of the entire crop in Canada. Canola production in Canada, just for those who may not be familiar with it, it occurs mostly in Western Canada. Large acreage of canola is produced there, principally in the darker soil zones, which is that darker yellow band. And that covers Winnipeg, where I am located, and the other major cities in Saskatchewan, Saskatoon, Edmonton region, and we have some production in Grand Prairie, and we have light production in other parts of the country, the lighter soil zones in the west, as well as a little bit in the east. But most of the production is in those darker soil zones where the weather, climate are much more conducive to high-yielding canola. The pie chart I've included here is the crops acreage planted in Manitoba on an annual basis. And canola covers about 30% of the acreage in Manitoba each year. Another 30% is wheat, and then the last third is made up of a whole host of different crops, soybean being the dominant one over the last 10 years, and it's displaced barley and oats. So we've shifted in Manitoba from a cereal canola rotation to a cereal canola soybean rotation, which may have implications with respect to protein content in canola. I won't speak about that much today. But hopefully I'll have some more news on that in the future. So moving on to our first data set, this is the data we collected from the Canadian Grain Commission through Veronique. It covers the years 2009 to 2017, and these are sample submission locations. So these are locations. These aren't the actual fields. We don't have that information. We only have submission location, which is typically an elevator or some grains buyer. And as you can see, it covers the canola growing region quite well. The data set itself, as I've already indicated, 2009 to 2017, we've only included Brassica napus hybrids for which we have more than 100 fields of data. We've classified the locations and collection points into 38 different crop districts, they're real crop districts. We're not making those up. And we have in the analysis about close to 10,000 fields. Seed protein values at the bottom are listed. The ranges are it's quite a wide range. The minimums, Manitoba and Saskatchewan, in Alberta are relatively similar, and they're quite low, which tends to indicate that there's an adverse weather event. Early frost may have caused that. Maximums are close to 30%. And the means, which the range I've provided for, is the means by year. We essentially in Manitoba have a range of about 4% from 19.6 to 23 point something, and in Saskatchewan, Alberta, about 2% range over those 8 years of data. Variety distribution is given on the side. In terms of varieties, the top 5 canola varieties that are grown in Western Canada cover about 40% of the acreage, and then everything else follows after that. So in terms of sources of variation, we did a very simple percent sum squares calculation, and we also indicated significance of those various effects. These are effects that we put in the model. We don't have a lot of explanatory variables in this case. We have year, we have district, we have genotype, and then the various interactions. And what becomes very obvious very quickly with the on-farm data is that we're not very good at explaining protein content on farm. The residuals cover about 70% still, so there's 70% of the variation is unexplained. And while many of the factors that we've included in the model are highly significant, they really don't explain a lot of the variation that we see. So from a modeling perspective, not great. But tells us that there are a lot of things that we can still learn. Now, why is that residual portion so high? Could be a couple of reasons. Number one, just inherent random variation in canola protein content, which there probably is a component to that. Canola is a very plastic plant, or Brassica canapeus is a very plastic plant, so there would be a fair portion of that. However, we would also, in this model, be missing some very important explanatory variables. And so part of that lack of explanation or lack of that explaining the protein content of the large residuals is the lack of explanatory variables. But nonetheless, it's very good to know what we can glean from the on-farm data. Now moving on to our next data set that we've done a fair bit of work with, and this is the Corteva AgriScience small plot experiments. The data years range from 2017 to 2020, so 4 years. 65 different environments, and they're distributed relatively evenly across the 3 prairie provinces. We have about 427 genotypes in this study and about 16,000 total observations. So a very large data set. Distribution of those data sets or of the locations is through the prime canola production area, so Manitoba, Saskatchewan, centered around Saskatoon and Alberta. We're not covering some of the lighter canola production areas with this data set, but nonetheless, it's a very good distribution throughout our principal canola growing regions. So if we look at protein concentration within that data set, things look very different than they did for the on-farm data. In this case, we have a bit more information. Again, we have a year effect, in this case, an environment effect, which would be the location, a genotype effect, and then the various interactions. And the good news here is that we're explaining about 70% of the data with our explanatory variables. Again, the year effect is relatively small. The genotype effect, although highly significant, is relatively small, only explains 5%, and even the genotype by environment interaction is only 7% of the total variation. Importantly, most of the variation is tied up in environment. Over 50%, or about half the variation in this data, is tied up in the environment/location effect. And I'll talk about that in a second. There's still 32% of the variation that's unexplained. So as before, might be some random variation. It might be some key factors are missing. So let's look at the environment a little bit more closely and what is that comprised of. In the breeding world, we always talk about G by E interactions, or more recently G by E by M, and I've split up the E and the M into the 2 circles here. And the environment, though, is comprised of a whole host of different factors. Some of them we have real good control over, for example, the agronomy, which oftentimes is referred to as the M part, and others we have a little bit less control over, which would be weather, precipitation, temperature, solar radiation, etc. And then there's a bit of overlap because some of these factors are certainly interlinked. For example, Some of the agronomic factors, such as rotation, would probably or would affect organic matter, soil pH, etc. So there's a bit of overlap within those. But nonetheless, the environment component is comprised of a number of different variables. And we've taken as many of these for the time being out and added them to that data set from the Contraria Corteva small plot trials and conducted a partial least squares analysis from that, or using that data. So here are some of the weather variables that we've included with the PLS regression. And the first two variables are talking about or looking at days above a certain base temperature. So with HD, we have the number of days counted above 25 degrees, 28 degrees, or 31 degrees, different category or different variable for each one. In the next variable, we've calculated that as growing degrees, again with a base temperature of 25, 28, and 31. And for each of the variables in this weather file, we've actually created a count by week to maturity. So we have the total for each of these variables within 7 days to maturity, within 14 days to maturity, within 21 days to maturity, and we go back many, many weeks. Unfortunately, we don't have flowering data, so we had to base it on maturity information. Another variable we have included is the daily temperature range, and we've turned that into a categorical variable with low, mid, high, and extreme range in daily temperatures, as in daily high, nightly low. The categories are based on quartiles. We basically took the largest range that we could find, divided that by 4, and then categories classified them accordingly. Total precipitation for all the different weeks is included. Solar radiation, crop evapotranspiration was calculated based on Martel's model, and water deficit is included as well. And the ranges, if values are provided for the entire data set from emergence to maturity, just to give you an idea. We've also included agronomy factors in this PLS regression, and the ones we've included so far are seeding date, we had those, levels of fertility, and these are fertilizer applied. They are not total soil fertility. We haven't included the residual nitrogen, phosphorus, potassium, or sulfur levels, and we will do that in the future as we get that data. And we've also included previous crop in the model, but in the trimmed model that I'll present in a minute, we've excluded that. There seems to be a crop effect, but we're not quite sure whether that's caused by the unbalanced nature of the crops or whether that's a true effect. So it'll take a little bit more time to analyze. We also included some genotype effects. In this case, with the data that we had were continuous variables, which are days to maturity for all the genotypes, as well as plant height. And the genotypes themselves as a categorical variable with 427 different genotypes. So you can imagine that the matrix going into this partially squares regression was quite large. It had hundreds of variables across the top and 16,000 data points along the side, or 16,000 rows. Many columns, many rows. And then with PLS regression, we would trim that model to the most important factors in the end. So what PLS does is it reduces the data to as few latent variables as it can to explain the most amount of variation in our response variable. And this is what we found. Once we've got the model trimmed down to the least number of variables required to explain the most amount of protein percent, we found that all the variables that mattered had to do with environment. One of them was nitrogen fertility, so that's agronomy or management. But the other 4 variables that defined our 3 latent variables are related to just environmental conditions. Together, those 3 latent variables, which are essentially 3 linear equations, explain 27% of the protein content, which is actually quite good for a relatively small number of variables. The parameter estimates and standard scale data are presented in the table below, and they're quite important because being centered in scale data, they tell us the size of the parameter estimate actually tells us the degree of influence. So the most influential factor here was number of days above 28 degrees Celsius in the last 7 weeks before maturation of the crop. So late during development. And that factor had the largest parameter estimate, therefore was the most important. And it was positively associated with protein. More hot days above 28, greater protein level. And fertility was the next most important one. And again, we haven't got total soil nitrogen, so level of nitrogen fertility positively associated with protein. Does make some sense. Precipitation in the last 7 weeks of before maturation was also important, and more precipitation meant more protein. What was also interesting in that period of 7 days, 7 weeks to before maturation to maturation was the daily temperature range, and the low daily temperature range actually had a negative effect on protein. And that tells us that warm nights during that period are detrimental to protein formation, which is quite interesting. Last but not least, a very small parameter estimate is solar irradiation until maturity, so that's season-long solar irradiation. We're not sure whether this is driven by latitude only and may be related to perhaps yield, and the yield and protein relationship, or whether that has something to do with more cloudy days versus less cloudy days. We will still need to dig into the data to figure that out. But this is our trimmed model so far, and explains, or at least tells us which of the environment variables that we've included so far are key to protein formation. Now, shifting gears a little bit, here's another effect that might be important in explaining what we see on farm level, and we typically wouldn't see this at the research level unless we design studies specifically for it. This is a graph, a frequency response of canola stand densities in farmers' fields. This is from a survey we did in my lab about 10 years ago. And we've got 206 fields here, and we've got plant densities in the range of 20 plants per meter squared, all the way up to 120 plants per meter squared. So very, very broad range. And the fact that canola emergence is highly variable is very well documented. And it's still one of those pieces that we need to, of the puzzle that we need to solve in the canola world going forward. Nonetheless, we have regional statistically significant effects, year effects in this data, as well as producer effects. Some producers always have lower densities in their field. Some producers always have higher densities. And the producer effect obviously is management, and that we can control. Now, how does that relate to protein, or what can we glean from that? Well, at the moment, myself and Dr. Shirtliff at the University of Saskatchewan are involved in this project where we're modeling canola yield in response to spatial arrangement. And we've got very low to very high densities of canola, as well as a number of different row spacings. And we're modeling the yield response. And yield tends to maximize at all locations right around 40 plants per meter squared. But we have much, much lower densities as well, particularly in the last few years when we have had drought conditions and emergence has been quite poor. I took this data, and the project was principally about yield, and I had Steve ship me all his samples, and we ran them through our NIR and looked at the protein plant density relationship. And what we see for a number of locations, not in all, but in 75% so far, is that we have this negative relationship between plant density and seed protein content. And over that range of 20 to 120 plants, in this particular location, I've modeled it, it looks like a linear relationship, and we see roughly a 1% or just over 1% range in protein content variation based on density alone. And of course, you see the picture on top in the top right corner. Those are the differences in plant morphology of a high density stand versus low density stand. The plant on the left is a stand density of 80 plants per meter squared. The plant on the right is stand density of 20 plants per meter squared. Very different morphologies, high plasticity in canola. And at lower densities, we shift the flowering period and we shift the time to maturation. And all that seems to have some effect on overall protein content. So this is very exciting because it may help explain some of the things we see on farm, but we would typically not see this in research experiments where we aim for much, much more uniform stand densities across our treatments. So that's what we have so far. In summary, I'd just like to reiterate some of the main points. Number one, there's a lot of variation in protein content in canola, even in that small range that we're working with, and a lot of it we can't really explain yet. Environment plays an important role, a very important role, and seems to be the overall driver when we're talking about multiple locations. Part of that environment we have control over, part of that we don't have control over, and we need to figure out and learn more about which of these parts affect canola protein content. Our PLS analysis showed that weather during late development, the last 7 weeks of crop development, were very important at this stage, and then fertility also plays an important role. And the other thing we've discovered through this other study that we've done on the side is that negative relationship between stand density and seed protein content that shows up at a number of locations and may also play a role in particularly what we see on farm, where we have these large ranges of canola stand densities. Going forward, this project is, as I said, we're about a year into it. We still have a fair ways to go. Going forward, we certainly are looking to do more work on the environmental variables in the data sets that we have. We'll build those and include more weather data, more soil data, texture, and fertility, and more agronomy data, tillage systems, et cetera, as we can build it in. And we may also use some additional statistical approaches for these for this analysis and look at perhaps random forest type modeling and some of those more AI-oriented techniques. And that's it for me for now. Here are some acknowledgements, including our funding agencies on the right that have contributed to the studies I've reported here, and the collaborators whom I've mentioned already. Thank you.